Now, it turns out that barrier proteins also play a role in separating segments along chromosomes that may be involved in different activities. And what we're going to look at is histone variants that are involved in forming the centromere. This is a stable, condensed structure, even when the rest of the chromosome is open. And this is the point at which microtubules and other things make contact. So we have normal nucleosomes and nucleosomes with these centromere-specific histone H3 molecules. And they have different properties. They interact with a sequence-specific DNA binding protein to form this centromere-specific structure with the H3 histone involved in the nucleosome structure. And that sequence-specific binding protein also binds to the microtubules that interact with the yeast kinetochore when you pull chromosomes apart after replication. So, chromosomal structures can sometimes be inherited, so that if you get alterations in H3 that recognize different regions, then you can sometimes get a second centromere at another site, or the centromeric site can move. So there are multiple processes in which histones are involved in addition to just packaging the DNA. Now, once you form this solenoid structure with H1, and you've got these really much more like closely packed beads on a string, during interphase, these are packaged further for the actual division process of the cell. And you add scaffold proteins to get this folded solenoid structure that is more compacted. And then, as we'll see later, there are more scaffold proteins. So to assemble chromosomes, we started with the naked DNA, we've gone to nucleosomes, we've gone to the solenoid structure. And to get to that solenoid structure, there are specific proteins that are involved, and they're called SMC proteins. And they sort of have this hinge structure, so they're able to latch around and, if you will, knit the DNA together. And they assemble into higher order structures themselves, as you can see here. They first bind, and then they form this 8-membered ring, and then those rings stack on top of one another to give the very condensed structure of chromatin just before cell division. So they form the folded solenoid, and then it condenses further, and finally you get the mitotic chromosome. So we know relatively little about these additional scaffold proteins that form the highly condensed chromatin that is characteristic of the mitotic chromosome.